is Sara, and I work as a research coordinator at Karolinska Institutet. And today I will talk, I will show how we had the help of HOPEsWorks to advance in cancer research, how we use it, especially for parallel computation. I will go through, what we do, a bit of cancer research and the challenges that we have faced. My position belongs to the Center for Cervical Cancer Elimination, so we do a lot of cancer research. I will show Biopipe, which is a bioinformatic pipeline that aims to detect all bacteria, virus, and fungi that are present in samples. In our case, we analyze human samples. I'll show you also how we run it in HOPEsWorks and how easy it is. And I will talk a bit about our present and future projects. So as Joachim already told us, cancer is a global health challenge that affects, not millions, but trillions of lives and millions of lives each year. The traditional methods for cancer research had limitations. We had pathology, radiography, cytogenetic. We could even have cell cultures. But we didn't understand with those techniques how the cancer was developing. What were the mechanisms that were undergoing down there? So there was a need to characterize the genetic and molecular aspects of cancer. And for that, we had next generation sequencing. When I started my PhD more than 10 years ago, and we were one of the groups, one of the first research groups that had a sequencing instrument machine. And at that time, we were very excited. We did our things in the lab. The protocols were very straightforward. We went to the instrument, sequence, and then we could get our data in a small USB stick. Sometimes we could even print the data in a piece of sheet and highlight with a marker the different mutations. Time went by, some weeks, some months, and the data was not a megabyte or a couple of megabytes, but it was doubling, triplicating. So we didn't have megabytes, we had gigabytes and even terabytes. So now it was impossible to print those out and do it manually. We needed IT, and that is something that we were not using in the lab. We needed parallelization, and that's something that we didn't know how to do. So we got stuck. Cancer research was stuck. The laboratory we could do, but then we were waiting months for simple analysis, and sometimes we didn't have the computer resources. So for going from 1 + 1, we had very difficult equations like parallelization. How could anyone do that? And from having this USB, we moved to a server and then to a cluster. And here's when HOPEsWorks gave us the solution. The challenges that we have in bioinformatics, and I wrote here data sharing and security. This is nothing that I'm going to talk about here, but we know it's a challenge as well. But from this lecture, from this seminar, it will be more about the complicated scripts. Bioinformatics is not easy, especially for people that do not know anything about IT. Apart from that, there's a lack of standardization and quality guidelines. We don't know which parameters or which tools we have to use. While on the opposite, when you go to the laboratory, the protocols are straightforward, and you know it's 2 microliters you have to add. Then it's 2, it's not 3, not 1. But here in bioinformatics, it seems that we are playing with the cutoffs depending on the data set that we have. And as I said, we have huge amounts of data. And for HEAP, what did we need to do? Well, we are to provide pipelines that are parallelized, automated, so it's not someone that has to be sitting there waiting for the process to finish and then click for the next. They have to be validated. We establish the cutoffs. We try them with different data sets to make sure that we are not making false positivities and not false negativities. Easy to use. Everyone should be able to run a bioinformatic pipeline. You shouldn't have a master of science in IT. And of course, open source, because they have to be accessible. We presented the HPV meta in our previous Bring Your Own data, because we belong to the Center for Cervical Cancer Elimination, and cervical cancer is caused by HPV. So of course we had to have a pipeline to detect HPV transcripts. But now we are presenting here Biopipe. Biopipe is a pipeline to detect bacteria, all viruses and fungi that are present in DNA or RNA sequencing data. In order to design and validate this pipeline, we use the whole TCGA database, which is a public database that contains more than 20,000 tumors. And we have also used it in different data sets that we have at Karolinska, like with cervical samples. As you can see, this is all the steps that Biopipe has. And the part on your left, which is the biggest one, is just the data pre-processing, because we have to make sure that our data is of quality. So there's a lot of quality trimming steps, human removal, and then at the end, when we have these high quality non-human reads, we do the classification with Kraken tool, which is like an open source and very well validated tool. As I said, as Alex said here, we're not reinventing the wheel, but we are taking the tools that are out there. All the authors and different scientists have used them. We are not there. We know that they are validated and work, and that's what we use. And after the Kraken, we have given some tools for diversity analysis statistics that are in R. So more or less, when we go into HubSource, this is how more the web page looks. This is the novel user interface. You go to pipeline here, and you click on open project, and here you have your project. Those three things are the most important things when you run a pipeline. The data you put in, that's the data input. The references that you give also, those are the databases, and the output. If you click on raw input or references, you will go here and you can upload your files. And this looks like a usual Windows Explorer, so it's quite easy. And when you upload them, you can see how they are uploading. Once I have my data input uploaded, it's just about running the pipeline. So for that, I go to Airflow, as Alex explained, and I can go here to Biopipe Kraken. And here I will see the different steps of my pipeline, and you will see different colors depending on how it was run. Well, in this case, it was success. That's why it's like dark green. But otherwise, you can see in which step it stopped and what happened. If you don't like this view, HubSource also offers a tree view. So it's up to the user to choose it. And of course, this is not something that is closed, that you have to use it as it is. You can always go to the code and tweak it if you want. We have like parameters that have settled, and we have our default cutoffs, and we have explained why we have those, but of course you can change them if needed. And the things that are interesting about Biopipe, well, actually all the bioinformatic pipelines, is that we have to know that all the output and the logs from each step are saved. It's not like in the laboratory when you mix two things together, you cannot say, oh, I want to go back. You cannot separate them. With bioinformatics, you can, so you can always go back and track and maybe change something. And the final output that someone gets is not just the microbiome profile, so it's not just about the bacteria, virus, and fungi that you have there in the samples. We also say, as I said, we also have some R scripts that calculate or analyze the alpha and beta diversity and differential abundance. And that is when people want to compare two different groups. Let's say that you have analyzed cancer tumors and healthy tissues, so you can see the differences between those groups. Here are some examples where we can see, well, the top 10 bacteria found in tumors and in mucosa, alpha diversities and beta diversities. I will not go very deep into that because I know is going to explain now a bit about it and give you some examples. And this is the differential abundance also, where we show like if we find the microorganism that is like more prevalent or more abundant in one group as compared to the other. We use the Biopipe in, there's one manuscript that is already, one paper that is already published where we use the Biopipe, and we have several others under submission, one of them including the Biopipe per se, where we explain the Biopipe. Now that this is done, what is next? So as I said, with Biopipe, we identify the bacteria, virus, and fungi that are known, and for that we use reference databases. But what happens with those that are not characterized yet? We always hear there are novel viruses that have been characterized, and so that is what we are aiming to. We will look for distant homologs and add those tools into the Biopipe so it can also identify those. And once we have that, we will move to machine learning with all the data to see if there are some patterns in the microbiome profiles that can be associated to cancer or to health status. And all this work is done by, well, I would like to thank, of course, Ainhoa and Dhananjay, which are the PhD students that are really hard working, well, not just PhD students, Master of Science in Bioinformatics and specialized in parallelization that are doing the whole work, and the PIs, Jim Dowling from Hopsworks and Joachim Dillner, and the whole HEAP team, of course, and EU funding. Thank you.